Good morning, my name is Lucrezia Mertova and today I would like to talk about eBCSgen, a software tool for biochemical space language. eBCSgen is a tool for development and analysis of models written in biochemical space language. The tool provides interactive model editor, model simulation, analysis of the model concerning PCTL properties, use the static analysis methods, and interactive data visualization. BCSL, Biochemical Space Language, is a rule-based language. The most significant advantage of BCSL is its ability to formally define the complex biological system, so it is fully computer-readable. But in the same time, it is easy to read language. BCSL preserved formal aspects needed for analysis, and for this purpose, the tool eBCSgen was created. The picture shows a simplified scheme depicting an artificial signaling pathway. This model is going to be our model for the demonstration of eBCSgen tool. There are two signals, one with a negative effect on the pathway, named block in the picture, and one with positive effect, labeled SIG. These signal transductions appear in two biological spaces. They start in the extracellular space and continue inside the cell. Let me write the definition of the running example in BCS language. I'm going to use the visualization of the model editor for this purpose. It is basically a simple text editor with syntax highlighting in real time and error detection. The model definition in BCS language consists of three parts. First of them is naturally the set of reactions. Because the BCS language is rule-based, we work with the abstract form of these reactions and we call them rules. The first two rules represent positive signal changes. The second one describes the signal traveling from extracellular space inside the cell. This action is symbolized by the change in the location part of the agent. Following block describe analogously the negative blocking signal. Another two blocks symbolize interactions of protein P1 with the positive and negative signal. And the last rules describe the relationship between the proteins P1 and P2. The second section of the model describes the initial state of the system. The indicator of this section is the keyword init, and is accompanied by the number of specific agents in a concrete location. Each rule is followed by the rate function, which expresses the speed of this rule. In the last paragraph are the values of the parameters from each rate function. In case some parameter is not defined, we call the model parameterized, and there is a possibility to run a model checking analysis on this model. The eBCSgen is developed as a command line tool integrated to Galaxy Server. It is a web-based scientific analysis platform focused on the analysis of large datasets. In order to be able to use this tool, it is necessary to learn the basics of the Galaxy user interface. The key components of UR are the following. On the right side, there is history of your computations. Data space, where all files and analysis results are available. This helps to keep track of analysis results origin, allows to easily chain tools, and share reproducible data. On the left side, we can see an eBCSgen analysis tools. The main functionality of eBCSgen can be found here. I will demonstrate individual tools later. On the top, you can get to published data libraries and histories available as reference data. There are also available files for this demonstration. One of the basic functionalities is, of course, simulation of the model. After selecting the adequate tool, you can work with this feature. First of all, the user is supposed to choose a model among the uploaded models from the history of computation. Next step is to choose the variant of the simulation algorithm. For this purpose, I choose the stochastic simulation, which is calculated by the Gillespie algorithm. The number of runs is selected to 100 and the maximum execution time is selected to 1. After execution, result data can be found in the history of the computation. Likewise, the editor, the simulation has its own visualization, which can be found right here. One can select the visualization of the data in text editor or the simulation plot. Text editor edits the basic CSV data, while in the simulation plot we can see the evolution of the agents over the time. Next feature of eBCSgen is the visualization of the transition system. Select the tool Transition System Generator. Choose appropriate model from the history and, optionally, you can define the boundaries of the final transition system. Also, there are advanced options like maximal computational time, maximal transition system size, and pre-computed transition system file. After the computation, the results are available in the history. The visualization of the transition system can be found here as a text editor or specific transition system visualization. This visualization is an oriented graph with the adequate probabilities on the edges. Self-loop can be either hidden or displayed, and the fire symbol represents the state outside the boundaries and can be hidden as well with all incoming transitions. Next feature I will show you today is PCTL parameter synthesis. Parameter synthesis can be performed only on parametric model, which means we have to load one. We go to shared data and then we access public libraries. In folder eBCSgen CMSB 2020, we can find one. Then we add it to our history. Finally, we can start analyzation. In history, we can see the parameterized model. Then we run parameter synthesis tool again, choose correct model, and enter a PCTL formula. PCTL formula expresses a property regarding the probability of an event to occur. If the formula has defined probability threshold, then the partitioning of the given parameter space to regions which satisfied, respectively violate the property is computed. Alternatively, we can set boundaries to the pre-computed transition system. Afterwards, the intervals for each unknown parameter in the model has to be specified. This is relevant only in the case when PCTL formula has defined probability threshold. In our case, it is parameter signal and parameter block, and in both cases, we set a beginning and the end of all allowed values. After execution, we can find the result data in the history. Result data in this case are of the format form.regions. Let's rerun the PCTL parameter synthesis and try another variant of the computation. In this case, we will exclude in PCTL formula the threshold, which means we will simply add the question mark instead of the value. But in this case, we do not define the intervals. If the threshold is not given, a probability function of parameters is computed instead, which evaluates the probability of satisfaction for particular parameterization. The output format in this variant is named storm.sample. This format can be visualized by another tool named probability sampling. Let's try it. We enter the recent storm.sample file, and then we have to define the previous intervals of the parameters. Moreover, for each interval, it has to be specified how many samples should be taken. These intervals of allowed values for each unknown parameter together form parameter space to be sampled. The result of this tool is an HTML file containing a graphical output of the sampling. There is a possibility to visualize this data as a text editor or to download this data. We can see the visualization of results of parameter synthesis. The left picture depicts partitioning of parameter space as a result of parameter synthesis with a defined probability threshold, 0.2 in this case. The right picture, on the other hand, depicts the sampling of the probability functions of parameters as a result of parameter synthesis without a defined probability threshold. Both pictures display the same parameter space for our example parameterized model. To sum it up, we developed a tool named eBCSgen. This tool is supposed to maintain models written in biochemical space language named BCSL. It supports simulation, PCTL analysis, and visualization of analysis results. It was implemented as a part of Galaxy platform, and it's available online on the following link. Step-by-step instructions of this tool are available in our tutorial.